All right. Welcome back to the symposium. We start with a session of talks. The first speaker is Anastasiia. She's going to talk us about applications of SWATH technology for extracellular vesicle investigations. Welcome, Anastasiia, please.
Good morning. My name is Anastasiia Artuyants. I work at the Hub for Extracellular Vesicles Investigation and Auckland Cancer Society Research Center here at the University of Auckland. And before jumping into the SWATH technology and how we can apply it to the extracellular vesicles, I would like just say a couple of words of introduction to the vesicles themselves. Extracellular vesicles, or also we call them EVs. Don't get confused when you do your Google search. There are some irrelevant EVs out there. These are non-sized particles enclosed in the membrane, and they are produced by every cell. But these are not just empty bags. They are full of active, bioactive cargo, such as nucleic acids, proteins, and lipids. They're produced by the cells and released by them, travel to the neighboring or distant cells before they get uptaken, and they release their active and functional cargo in the recipient cells. And the reasons why we care about them that much is that because we know that after they get uptaken by the target cells, their cargo, depends on the nature of the cargo, can be beneficial or detrimental to the target cells. And they become functional there, and they can influence the behavior of their hosts. Another reason why we care about them is that by circulating in the extracellular space, in all the biofluids of the body, they can be studied as the potential biomarkers, especially in the disease studies. And we have a lot of tools available to study how these vesicles look, how many of them we have in the samples. So for example, with nanoparticle tracking analysis, we measure their concentrations in our samples. We'll look at how they appear at with transmission electron microscopy or cryo-electron microscopy. We can look into the details of the surfaces with the atomic force microscopy, or look what spectra these vesicles produce with the surface enhanced Raman spectroscopy. And we have a lot of omics techniques available at the moment to look into the details in the cargo of these vesicles. In our projects, we were interested in the protein content of these vesicles, and that's how we get to the SWATH, or sequential window acquisition of all theoretical mass spectrum. This is one of the methods of the data-independent acquisition, which means that it records fragments of ion spectra, regardless whether they are present, whether their precursor ion is detected or not, which is in the opposite to data-dependent acquisition, where the peptides are specifically selected for fragmentation based on their detection in precursor ion scans, which means that in this case, we are losing low abundant proteins that were not detected. So how the method works? First of all, this is a label-free method that theoretically allows all the peptides in the sample to be identified and quantified. It divides the whole mass range into the several sequential windows, and all the precursors ions in each window get fragmented sequentially and quantified, which overcomes a loss of low abundant proteins, again, which happens in data-dependent acquisition. And it also avoids the drawback of only targeted interesting proteins, which usually happens with the selected reaction monitoring. And the method has several undeniable advantages. First of all, it's how simple the data acquisition is. Once you set and optimize your settings for the one sample size, you can robustly transfer it to other samples. Secondly, it's multiplexing and the breadth of peptide detection. Everything is simple here. Increased number of quantified peptides directly leads to the more precise and more accurate protein quantification. This method is very reproducible, which is very important when we get to 100 samples per study. And another interesting and good thing about the SWATH is that we can always go back and add some new data to the already constructed library, and there is no need to rerun the samples or do the acquisition again. And why we think SWATH might be a good fit for extracellular vesicles is because, first of all, most of our samples are complex, especially when they derive from the biofluids. We usually rely on the accurate and precise comparison, qualitative comparison between the study groups, and we are often interested in these low abundant proteins present in our samples. And there are some beautiful examples how this method was already used for different types of vesicles. But today I would like to talk about a couple of our projects. So first time when we applied SWATH method was part of my PhD study, where the design and research questions were pretty straightforward and pretty simple. So we had two bacteria that represented different opposite spectrum of the vaginal health. We had bacteria that is known to be present in healthy women, Lactobacillus gazzeri, let's call it good bacteria, and the other one that known to be causative agent of bacterial vaginosis, Gardnerella vaginalis, and let's call it bad bacteria. We isolated vesicles from two bacteria and we further purified them. We took samples from each of the group for the proteomics, and we were just looking for the protein profile in each group and whether there is some specific proteins are being packed into these vesicles or not. Performed proteomics, and the first we looked at the heat map, or how the samples clustered together or separate from each other. And the interesting thing that we noticed with the clustering pattern was that there were two distinctive groups of proteins present, for example, in the cell group that formed two big clusters. And the pattern of the vesicles cluster was almost invertible. And when we performed the gene ontology analysis, we saw that these were mainly membrane proteins, where exactly the proteins that packed into vesicles come from, and the rest of them. We performed the pairwise comparison of the proteins from the cells to compare them with the vesicles. And this is an example of a good bacteria. And the proteins highlighted in the table are the most interesting ones that we found here. So first of all, this is the enormous fold change of the proteins packed into vesicles in comparison with the parental cells. And again, proteins that are highlighted are the main antibacterial agents that these particular bacteria use to compete with others in their ecological niches. We did the same comparison with the bad bacteria, and again here I'm highlighting the proteins that were highly abundant in the vesicles in comparison with the cells. And these ones are mainly pore-forming toxins that are used by these bacteria to specifically lyse their target cells. So they use these proteins to attack the host and neighboring bacteria as well. So to conclude this part, because we used SWATH method, we confidently identify thousands of proteins for each bacteria, while only a few hundreds were reported before. We found highly abundant proteins in the vesicles in comparison to the cells that are directly related to the biological functions of these bacteria in their ecological niche, which gave us a target for the further functional study. Our next project was more sophisticated with more complex questions. So for this study, we had 3 breast cancer cell lines that represented the aggressiveness of this cancer. So we had MCF7 somewhere on this side, less aggressive, BT474 somewhere in the middle, and MDMB231 being more aggressive, and the tumor of this cell line will have the worst prognosis. But we didn't grow these cell lines on the flat plastic. For the isolation of vesicles, we use the bioreactor system. So this system is basically the size of the normal T170 flask. It is divided by the semi-permeable membrane over here into 2 chambers. On the top chamber, we add culture media. The cells grow at the bottom chamber. Here's a bigger image of the bottom chamber. So the cells grow in here in more physiological conditions, and they mimic how the tumor progresses in the body. They get nutrients from the top, and they release vesicles in here. And we collected vesicles from this chamber. And we successfully grew these systems in a parallel for 3 cell lines for about 4 to 6 months. Because the system is closed and we can't directly assess what's happening inside, we thought that maybe by characterizing the vesicles, we will be able to tell about the health of the system itself. So we were feeding our bioreactor once a week, and the feeding cycle is shown here. And we were collecting vesicles twice a week. So first question for the proteomics was that whether this feeding cycle may affect the protein profile of the vesicles. If we feed only once a week but collect twice a week, can we get some starving proteins or some indications that our system is unhealthy? So we collected the vesicles for proteomics at the early stages of the seeding, somewhere in the middle, and by the end of the life of the bioreactor. We performed proteomics and first analyzed the heat map that we got. So first of all, we saw that samples clustered together, again, in a similar way as they represent the aggressiveness of the cancer, with the more aggressive being separated from the other 2. And it was also confirmed at the PCA plot. Here we started wondering if there is a set of proteins that present consistent across all our samples and not changing over the lifetime of the bioreactor. And another important question we asked, if there are proteins that are changing over the time and if this feeding cycle may affect them. So first of all, we identified the set of the proteins that we called the core proteins. This is the ones that were present in all the samples and remain stable despite the stages of the bioreactor. And we performed the gene ontology analysis of these proteins. And we saw that the main source of these proteins was identified as the extracellular exosomes or membranes as expected. We looked at the biological processes of the proteins we found, and these are mainly processes that represent, first of all, the cell lines that proteins derived from, and also typical extracellular vesicle processes. Next, we identified the proteins that were indeed changing over the time, so from time points or from feeding points. And we performed the gene ontology of these proteins as well. And what we found, to simplify this picture, is that these proteins were not overlapping with the previous identified ones. And we confirmed it with this Venn diagram. So what is important, that our core, our set of proteins remains stable, and it's not overlapping with a variable one coming from the different cell lines. So we have started wondering about the source of variability and changes in these groups. And it's turned out that our initial idea that the feeding cycle affects them was actually not. And what was affecting these proteins is that the beginning or end life of the bioreactor. And we are looking in the details at these proteins at the moment to identify if there is a biological reason or these proteins are just less stable than the rest. To summarize this part and overall, again, by employing the SWATH, we identified thousands of proteins. We found the set of core proteins that remain stable. And again, we can use it as a readout or indicator of the health of the bioreactor system itself. And overall, the proteomics and SWATH in particular is a powerful and useful tool in our field, and it finds more and more application with a project similar to that. I would like to acknowledge MASH for organizing the symposium and inviting me giving a talk about our project today. Special thanks to Martin and George to support and help with our projects at all the stages: planning, troubleshooting, analysis. Thank you so much. I would like to thank my heavy team and my colleagues who helped with this project over the years, and also Breast Cancer Foundation New Zealand for supporting this project with the Technology and Innovation Project Grant. Thank you.
We have time for only maybe one tiny short question. No? Okay, maybe we can move on. Thank you so much, Anastasia. We're going to invite now Andrew there. Let me put. Andrew is going to talk us about space, the final frontier, the spatial localization of berry fruit metabolites using MSI. Welcome, Andrew.
Thank you. Kia ora, everyone. Yes, as just said, I'm going to talk to you today about plant science, so please don't leave the room if you're a mammalian biologist. It's going to be talking about some work we'll be doing in collaboration with Gus Gray using MSI to look at metabolites. Okay, so I was told I had to do a Star Trek reference if I was going to use Final Frontier in my talk title. So this is it. I resist the temptation to put Spock ears on Gus. So these are the people I'm working with. I like to thank them straight off, because without any of the work I'm going to show you, none of this would have happened. So as I said, I've been working with Gus at Faculty of Health and Medical Sciences, and Nick, who some of you might know, who's just got a job in the real world, so ex-University of Auckland now, so congratulations, Nick, who drives the FTICR, and he's definitely a guru on that, so he's been invaluable as well. And of course, our data guy, George, who I can see sitting there, who crunches the numbers and does the really important stuff in terms of the bioinformatics. Okay, we haven't been using the USS Enterprise for our work. It's something probably a little bit more expensive than that. The Solaris FTICR, which is based up here in the Iron Innovations Lab. And this is a very, very impressive machine, has very high mass accuracy and mass resolution, which is really essential when you're doing MALDI work, because we're doing in situ localization. We can't do any LC separation. So mass accuracy becomes really important. Okay, so to convince you, why would you do all this hard work? Mass spec imaging is quite a lot of, it's very hands-on and it takes quite a lot of laborious setup work. Whether or not you work in an animal system or in a plant system, the theory is the same. We all know that mice just aren't chunks of mince, and plants, hopefully you'll know, aren't just cold store. Okay, these consist of specialized cell types and cell structures within both, and potentially within those specialized cell types, you have specialized metabolomes and you have specialized proteomes. So being able to localize these is very important. And when we traditionally do this, we grind up a tissue, we push it down LC-MS, and we know what's in there, but we don't know where it came from. So to do this, well, first of all, I'll talk about where the money came from. So this is very important. I should thank our sponsors, which is the MBIE Endeavour Fund program called Filling the Void. It's behind the little window there. And we're trying to understand the phytochemical production and the flesh of blueberries, which are the big ones on the left, and their close cousins, which are called bilberries. Okay, blueberries you find in the supermarket, bilberries you won't. These only grow in northern Europe, and they're interesting because they have red flesh. Okay, so we're trying to create a hybrid between these two. The 1% of the blueberries, the healthy part, which is the skin, we want to fill up the other 99% with basically these health compounds, which are anthocyanins, which have this kind of amazing health halo around prevention of a number of lifestyle diseases. So back to the bilberries, they're the small ones that grow in Scandinavia. They only grow wild, they're not cultivated. They have a very high level of flavonoids, particularly anthocyanins in the flesh. And some might say they are the most significant thing to come out of Scandinavia since these guys. Okay. So what are we looking for? We're looking for flavonoids, okay? So for a plant biochemist or plant chemist, these are a really important group of compounds. These are phenolic compounds that the plant produces usually to defend itself against pathogen attack, or in the case of anthocyanins, the red ones, to say, you know, come on, eat me if I'm a berry, or pollinate me if I'm a flower. So this is the kind of diversity you'd expect to find within a bilberry fruit, another group of compounds. And these are not weird things that sit on a chemical shelf. These are all part of our diet if we're eating fruits and vegetables. So really important to understand how they're made and what effects they have on us, as well as the plant. Okay, so as I've said, MALDI imaging is quite labor intensive. And this is the kind of, I guess, the 4 big S's of it. A lot of the work's in the sample prep. So to start with, you need to cut up the tissue. You need to slice it. We use a cryostat. So we use by freezing these tissues, freezing these berries. And unlike people who get the luxury of working with animal tissues, which are high in protein, plants are low in protein, high in water, and water is not your friend when it comes to freezing it in a cryostat. It's like sectioning a glass marble, basically. So tricky to work with, but after a bit of work and a lot of swearing, I managed to get some reasonable sections in this example way. So the section gets done, it gets put on a slide. Okay, then this medieval apparatus on the far side there is our sublimation gear, and you apply a matrix to your sample. Okay, so the matrix is really important for MALDI because that's what absorbs the energy of the MALDI laser and transfers it to the analytes which are in the tissue. Okay, you can either spray it on, we've got an automated sprayer there, or in my case, we use sublimation. So we use this vacuum jar here, and inside there is our matrix, which gets heated, vaporizes, hits the slide, and condenses, and puts on this lovely kind of layer of matrix over the top. Nice and thin, no water involved. It looks pretty low tech. The glass sandizer sitting in was cost me $4.99 from Kmart, so it's not all high tech equipment. That's whatever works. Okay, so then it goes into the very high tech FTICR. And basically what happens is a laser rasters across the sample, shooting at 50 micron apart. So this is the resolution I'm looking at. Theoretically, it can go down to 10, but that's kind of just on the label that the manufacturer is selling, I think. So this is still really high res in terms of looking at a whole bilberry fruit. Laser beam fires in, hits the surface, and basically transfers that energy to your analytes, and then they basically fly as they're charged, and they're separated on the basis of mass to charge ratio. So in the ICR, they're basically, it's very complicated. Basically, they're accelerated to an ion cyclotron resonance, which allows them to be separated on mass to charge based on how quickly they spin. So we then put it through a computer program called Flex Imaging, which reconstructs at each point on the tissue, 50 micron spot, the spectra. So it's pretty spectacular in terms of the amount of data it's generating. And you can see there that's an image of a particular compound called chlorogenic acid, very common phenolic acid in plants. And that's kind of like a heat map of where it's highest. Okay, so the red and the green colors are the highest concentrations, and the blue is the lower concentration. So I hate to chuck a table up with a whole lot of numbers on, but I'm going to chuck a table up with a whole lot of numbers on, just to show you what we found. So we had a target set of metabolites based on our LC-MS. So what we also did with these tissues is peel them, and I don't know if you want to try reconsidering your career options, but one of them is to sit in the lab all day with things about the size of a pea, trying to peel it with a scalpel blade and liquid nitrogen. It really makes you wonder why you did science. But we did that, and we got LC-MS data as kind of to support our moldy imaging data. So these were our target set of metabolites, and we've got pretty much all of them. So it was pretty impressive in terms of what we're looking for. We got out anthocyanins with the red bar, class of compounds called flavonols with the yellowy bar, the tannins, which are in brown there, and the phenolic acids in green. So the other thing to point out with this is we've got really good mass accuracy. So, well, I think it's good. Anything less than, I think, 5 ppm. So this is the deviation from the expected exact mass that you get. It's pretty good for moldy imaging. And I think we're all less than 3. So very close to what we'd expect. We managed to also do MS-MS. So this is another level of validation with the parent fragment is fragmented into fragment ions, footer ions. And then we can see basically and get that extra level of validation to see if these compounds are correct. You can only really do this in the more abundant ions in the case of moldy imaging stuff, but it seems to work pretty well, particularly for the anthocyanin. So just another level of validation that what we're seeing is what we think we're seeing. One small downside is you see this here is this hexoside. Now chemists hate the word hexoside because it can refer to a number of glycosides. And the annoying thing about plants is they produce lots of galactosides and glucosides. So these are sugars attached to the flavonoid backbone. And they have the same exact mass. So you can't differentiate, as I said, there's no LC separation, no liquid chromatographic separation. So you can't distinguish between those. So we've got to call them hexosides. But that's just a small price to pay, pretty pictures you get at the end. So I'll just run through a small set of pictures because it's just like you found the album. You're always really interested in it. Sometimes people looking at the album aren't that interested in it. So this is my family album. These are the 3 stages of development of the bilberry fruit. Chlorogenic acid, which is just very common. It's pretty much in every plant, so it's a good thing to look for. No one knows what it does. In terms of the bilberry fruit, you can see it's very high early on in the skin, and then it kind of drops down over development as it gets more mature. Okay, and this is kind of characteristic of a compound which has been produced to defend the plant or to screen out the UV. Similarly with procyanidins, these are part of the tannins group, the condensed tannins, things that are in red wine, probably familiar with. Very high in seeds, around the seed coat, but hard to see on the screen, much nicer on this up here. But basically around the seeds and the seed coat and declining with development. Because the plant is trying to defend its seeds, it's producing astringent beta compounds to protect them. And as it gets older, it says, I want to be eaten, so I want to make sure that these compounds, which are antefeedants, are reduced in these areas. Some of the other trends are basically, there's a big class called flavonols. We expected them all to be pretty much the same. They weren't. Some increased over development. You can see there coming on the skin in the middle stage at stage 4, while others decreased, such as this glucuronidine. These are very, very similar compounds. So very surprised to see they have a different accumulation pattern, which tells us a little bit about the biology of the plant. So you might be able to see it on this, but these little tiny little dots in there, which are the vascular bundles, which is the piping of the plant, which brings in the sugars and the water. And you can see them being produced in there too. So we think this is likely to be acting as an antimicrobial against compound. But again, a lot of these things have never been visualized in any plant, let alone in a berry fruit. So this is all kind of new stuff. The other surprising thing, well, these are red flesh fruit. When you cut them in half, they just look red. You know, there's no particular interesting pattern going on with the anthocyanins. Instead, when you look on the malva, you can see there's quite a strong gradient in concentration. You can see it particularly on the delphin and hexa side, and the malva on the right there, on the outside going into the fruit. So even though these are red flesh fruit, they're still getting signals from the external environment, which is telling them to produce anthocyanins, and it's basically a skin to flesh gradient still. So there were some different patterns across development. You can see this one here is basically peaking in the middle stage, with most of them peak at the late ripe stage of development. So it was interesting to see that this gradient effect, and this is telling us a little bit about how the plants produce the anthocyanins. We now think it's related to a hormone, which we know is produced from the skin and diffuses into the plants. We think this is co-tracking and driving this gradient. So it gives us a few clues about how to create this situation in blueberry. These are kind of some bonus molecules. We weren't looking for sugars, but it's interesting to look and see in terms of plant physiology. When plants are growing, you've got a young fruit on the plant. The sugars are coming from the leaf and being loaded into that young fruit, and that's mostly sucrose. You can see right in the middle there, in the middle of the plant, sucrose concentration is the highest. That's what we call PTL, which goes down into the middle of the fruit. And this is where the sugars, the sucrose, is coming in and being unloaded. As it matures, you can see there far right there, the sucrose pretty much starts to disappear. That's because there's an enzyme in there called invertase, which converts the sucrose into glucose and fructose, which is represented by these hexane sugars here. So these basically have the opposite pattern expression. As the sucrose is converted into glucose to make the fruit sweet, so we can enjoy it and eat it. Okay, just to finish off with, I'll talk to you a little bit about some segmentation analysis we did. So I had quite a small target set of molecules. It's like 25, 26 compounds, but mass spec imaging generates, hundreds of NZ values and potentially thousands of spectra, depending on how big your sample is and what sort of resolution you're sampling at. So we did some untargeted segmentation analysis. This is to create like a phytochemical map without kind of looking at skin and flesh and the bits we know about. What do the chemical signals tell us? So this is kind of, to put it simply, it gets all the spectra and groups them together in terms of their similarity. And then those groups are kind of plotted out as kind of color maps on the fruit, on the image section. And you can do this, as I've said, without any prior knowledge of what's going on in terms of the structure. So we just said, what's the chemical map during development of the bilberry? So you can see at stage 3, the early stage of development, it's pretty much one zone. Okay, there's one zone of chemical compounds, which is the green area. But you can see down the bottom here, you're already getting the transition, the stage 4 zone starting to appear here, which is the blue. By this kind of midpoint, this is kind of like the Veraison period, which is basically switching from the green to the red fruit, the ripening process. You've got a snapshot of the fruit in transition where you have the stage 4 chemical compounds there and a little bit of the immature stage 3 zone. And you've also got the late stage, which is the stage 5, the ripe stage chemical compound zone appearing on the outside as well. So we've learned a little bit about the way the fruit ripens here. We know that the stage 4 is basically contains all 3 stages in a kind of a hybrid. And we've learned that the transition to ripening in terms of chemicals starts from the outside of the fruit, from just inside the skin, and moves inwards. And we've done this in positive mode as well and found a similar pattern. So we think this is a pretty good indication of how the fruit business. And this is all new stuff because no one's done this before. No one's really understood spatially how a fruit ripens in terms of the chemicals it produces. Okay, so I'll just wind up a little bit by saying that the way with the mass spec imaging I've done for this has kind of changed the way we look at plant metabolism and the way we look at plant metabolites. As I've said, we can get a fruit, we can pull it apart, we can peel it, we can pull the seeds out, but that's about it, in terms of trying to get structural definition. So we can create as many graphs as like this as we want. We get concentrations in the skin, the flesh, the seeds. Okay, that's great. And that's kind of how we're working up to now. But we can now work on a much more refined level and hopefully get phytochemical maps of the fruit at individual locations. So instead of looking at a fruit as a whole, what's actually happening inside the fruit at a very high resolution. So this is a chlorogenic acid again. It's just one I picked out. It basically shows you where it is in the fruit, and the height of the peaks on this 3D plot is relation to the basically the concentration of it. So we've done a pretty simple data set. It's a development series, but imagine being able to do this with some environmental stress, with pathogen attack, with treatments, with sprays, with herbicides, all those kind of things which are going to alter our metabolome. We can visualize in 2 dimensions, or at least in 3 dimensions if we do this. Okay, so I'd like to finish my talk by thanking the people who have been involved. Us, Nick and George, a couple of people from Plant and Food, and I haven't put them up on the slide, but of course our funders in the IE should be up there too. I want to keep working. And I would like to thank you too very much for doing this. Cheers.
We have a few minutes for questions. Anybody? Yep.
When are you going to stop working on this?
Some of the images you showed, I think for some of the anthocyanins, you had high concentrations in the skin, but then you had that little spot in the center as well. So how do you explain that? Is there sort of transport between the 2 locations, or independent synthesis, or are they different molecules?
Good question. We don't think there's any transport of these compounds. They tend to get localized into the vacuolar cell, which is like a big bag in the middle of the cell that stores all these compounds once they're glycosylated. The little bit in the middle is the placenta, which is basically getting all the signals from the plant, most of the hormones that trigger vitamin, that trigger anthocyanin production, that trigger flavonoid production, come in through that piping. Okay, so what we think there is basically the signals, such as there's a hormone called abscisic acid, which we think is associated with anthocyanin production. We think it's a reaction to the signals which are coming down, and it can be sugars and it can be hormones. So it's a localized area where that signal is received into the fruit, I guess.
Thanks, Andrew, for the talk, sir. I think you just touched upon the last sentence about the environmental factor. Do you look at the soil factor as well, and the effect of the global warming on all this?
Yeah, we are working on plants, which, because one of the downsides of global warming is increased temperature drops anthocyanin production, so the fruit becomes nutritionally less valuable. So we haven't done any heating studies. We're working on a UV light study at the moment in terms of UV light quality, and not just sort of measuring metabolites, but measuring where those changes take place. So we're getting some samples sent from Norway in a few weeks' time that we're going to hopefully be able to do this at some stage, maybe. If they survive the transit from pretty much near the North Pole, yeah.
I have one question, Andrew. That's a fascinating talk, but why did you choose the bilberry, non-commercial berry that's only found foraging in Scandinavia?
You know how it is for science. You do something that works. So we did this because basically the bilberry is kind of our model for what a red flesh fruit should do in terms of vaccinating. We're doing it also, I started off doing it in blueberry, but at the time I didn't have access to the FTICR, so we weren't getting great results from it. So we switched to bilberry because it's smaller and easier to work with too in terms of sectioning. Got that, but we have, that's in the pipeline, is to repeat this exact art. Then we can compare basically the blueberry, which has white flesh, the bilberry that has red flesh. Yeah. So yeah, it was just logistics that we started on this one. This is taking a long time.
I have one more question. You're gonna try this with different or another fruits? Like this is tiny, right? So you slide it, and what about bigger fruits types like a kiwi or maybe I don't know.
Watermelon. Yeah, my boss asked me the same question. Asked me, could we get an apple on there? Like apples. So how many different bits do we have to do? That's a good question. That's another reason I'm working on berries, because they're small enough on the slide. Apples, most fruits start off small. Apples will start off, the development stage starts off small. Once they get big, the cells expand and they get much bigger. So I've seen this done on ripe fruit, but they tend to look at targeted regions. So you need to have some idea to start with where to look. So you can dissect the fruit down if it's an apple or a watermelon and do that. But yeah, you can't obviously fit an entire watermelon into a still expected to work afterwards. Yeah.
All right.
Cool.
Thank you, Andrew.
Thanks so much.
Our next speaker,
Failed with us.
it's Daniel.
Oh.
Sorry. We don't have your talk in there. Okay, so Daniel, he's gonna talk us about prospecting New Zealand fungi for novel antimicrobials using LC and GNPS molecular networking. Welcome, Daniel.
Hi, so I'm Daniel. I'm a PhD student in Susie Wild's lab at the Grafton campus. And one of the projects that we're working on is screening a collection of New Zealand fungi for novel antimicrobials using LC-MS and GNPS networking. That's global natural products, social molecular networking. So you might have heard about the antimicrobial resistance crisis. There's a lot of common pathogens that are evolving high levels of resistance to the antimicrobial drugs that are currently available. So things like MRSA, but also there's fungi becoming resistant to antifungals. And it's a big problem in a lot of hospitals. So I've just got this quote here from Margaret Chen, who was a former director of the World Health Organization, talking about if this trend continues, there's a danger we'll be heading into a post-antibiotic era in which common infections can once again kill, and a lot of surgical procedures might become too dangerous to perform. So we're trying to find new kinds of antimicrobials that help get around that problem. And fungi are historically a proven source of antimicrobials right back since the days of Alexander Fleming. And two of the best classes of antibiotics we have, for example, penicillins and cephalosporins, originated from fungi. And they're also a source of novel antimicrobials. So there's a lot of fungal species in the world. A lot of them are metabolically underexplored. And there's also been studies looking at sequencing and showing they have this untapped genetic potential of stylophyte gene clusters and that sort of thing. And they're even finding new compounds from known species like penicillin. So our fungi in particular, we get them from Landcare Research on Ngāti Whenua. This is the international collection of microsuppliers. That's Bevan Ware, who's who manages the collection. So it's people, often it's fungi that bracket fungi that grow on trees or decaying wood and that sort of thing. And they send them to us and we grow them on agar plates like this. And our original workflow, we weren't using LC-MS, it was like an NMR based workflow. So our process was we first grow them on the PDA, we test them for activity. If we found active fungi, we'd grow up 100 plates of them, freeze dry them, get them extracted in chemistry. Then they would fractionate their extracts on a column and send us back the fractions. So we get a crude extract of maybe 4 fractions, and we test those for activity. And if we found an active fraction, then they would subfractionate them and send us the subfractions. We test those for activity. And we just keep doing that until we purify the active compounds. And during this time, they're trying to solve the structure using NMR. And then the final step is to de-replicate compounds using the databases that we're literature searching. That's to find out if you found a novel compound or something that was already known. This is just a standard bioassay guided fractionation method, which is works, but it's kind of slow and labor intensive, and it often leads to the rediscovery of novel compounds. So we have found several novel compounds, but we've also found a lot of novel compounds. So we've been doing this for a while, and I was also reading some articles about this thing called LC-MS, and people encountering the dereplication problem, saying that LC-MS has advantages that might be useful for us. So it's ideally suited for analyzing complex mixtures like our fungi extracts. It has a very high sensitivity especially compared to NMR, which in our case allows us to work with smaller volume samples that we can grow more quickly. So for our NMR samples, if it's in liquid culture, we'd have to grow up like 1 liter or 2 liter flasks, which can take a month to reach full growth. But with the LC-MS method, I can grow them just like a 1 mil volume for like a week, so we can get higher throughput, and it may allow early stage dereplication. Also, the bioluminescent superbugs group, so we're working with glowing bacteria that have been engineered to glow, and we're just using that as a proxy for other metabolites that become active. So the more bacteria you get, and happier they are, the later they glow. And we can image it's a 96-well plate where we've done an extract test, and we're just doing serial dilutions to see like what's the lowest concentration we can go and still stop the luminescence. So we're just using it as a rapid sensitive assay for detecting antimicrobial compounds in our samples. And that's just the basic light mechanism. So chemical reaction, we don't need to add any substrates, they just glow constitutively. So this is when I started adapting our methods to include LC-MS. It starts the same, we just have to get on the air plates, and we use a little biopsy punch to transfer plates of mycelium into the 24 plates. And then if it's liquids, then we filter those and do some SPE and some LC-MS. And while we're doing the LC-MS, we also collect our fractions into these deep well plates here. We made our own fraction collector, which I'll show soon. And then we can transfer samples from the deep plates into these shallow black plates. And then I can make like 3 or 4 replica plates of this, depending on how many bacteria I want to test against. I add in the bacteria and incubate them and monitor the bioluminescence over time. So in this case, we're looking for any dark wells amongst the background brighter wells, so this fraction is active. And we can also do high resolution LC-MS on the active fractions. So we didn't have our own fraction collector, so we decided to make one based on this design from Bruno Vidal that our technician found. Video, but we modified it for use for our deep well plates because the movements needed to be more precise. Okay, so this is just our LC-MS stuff in series lab. So we just got a flow split at this, we arrange it so that about 10% of our flow goes to the mass spec and 90% goes to the fraction collector. That's a deep well plate that I was talking about. This is the Lego Mindstorms controller. We've got a flow rate of 1 mil per minute, and it moves just automatically every 30 seconds. So we're collecting, that's a bit cheeky, but we're collecting about 0.5 mil per well, just see the drips coming out there. We're lucky because our technicians into woodworking and Susie's drawn to Lego, so they have the skills to make it. Yeah, so the first fungi that we tried to test this method with was penicillium expansum, partly because it had very strong activity in our initial screening assays, and also because it has a lot of known metabolites. But some of that just grows on like apples and things normally. And our 2 main questions were, can we detect activity from the HPLC fractions that we're collecting? So we're not loading that much, we're only loading like 40 microliters, so it have to be quite potent probably. And can we identify the active compounds just using the LC-MS data from these small scale samples before we go all the way in? So this is the results from the penicillium patulum sample expansive sample that we ran. It was active against 3 out of 4 of the bacteria that we tested. So it's well B9 there, it's active against S. Staphylococcus. And in this assay, we can also, this is bacteria as well, so we can spot them out into agar plates and see where the colonies form. So it was bactericidal in a couple of those samples, storing colony formation. So it's quite a potent. And the so fracture 21, there was a retention time around 10 minutes. So then we go back to the chromatograms and we look at the peak at around 10 minutes, which is about 157.05 Z. So what could this be our active compound? So we get the monoisotopic mass and search that in databases, and we can filter some of these databases by species or genus of fungi. And one of the ones we use is Natural Products Atlas, and it found the top pick was this compound called desoxypatulinic acid with that accurate mass. But then we did some literature searching, and that compound is inactive against E. coli, which didn't match up with what we were seeing before. So maybe that's not the right compound. But it's also a metabolite of another compound called patulin that is highly toxic to bacteria, including E. coli. So we thought if it's making DPA and DPA is a metabolite of patulin, then it's probably also making patulin. So maybe that's our active compound. And then we're looking for this mass in our LC-MS data. So this is the full run of an hour. That's the extracted item, just between 150 and 160 Z. We've got a peak here at around 10 minutes. That could be our DPA peak. And then there's a smaller peak next to it, which could be patulin. So maybe that's what's giving us our activity. But it could be that most of it, of the patulin in our sample had already been converted to DPA by the time we did this. So, but we were also able to run just the active fraction by itself on some of Martin's high-res machines. So we've got a reference MS2 spectrum for patulin at the top. We just found that on the internet. And then our spectrum here, which so all like the peaks in the reference spectrum did have corresponding peaks in our spectrum, so it's consistent with the theory that we do in patulin. Plus we've got some extra peaks. This one's like our sample wasn't that's pure. But one thing we did find, because we tried to do it first in positive mode, and that did produce a very nice looking spectrum, but we got more MS2 peaks when we did it in negative. So to confirm that, we did grow up a large scale culture in the MR, and we detected patulin and various other metabolites of patulin in the MR sample. So we showed that we can identify active fractions and dereplicate known compounds, at least when they're well known and quite active. And we thought that ideally we would be running our samples in both positive and negative ionization mode, particularly because it's untargeted screening and we don't know what we're looking for really until we find it. And the next step was we wanted to try out this global natural product. This is the GNPS networking. So you can upload your MS2 spectra from like entire LC-MS runs, and then they'll compare it to compounds that they've got in their databases, and then they'll try to align the spectra to align the compounds in between the compounds that you uploaded as well. So here they've got the masses and some circles, and then blue represents unknown compounds and represents compounds that have matches in their databases. So it's found it's networked like some of the compounds that we put in. A lot of these couldn't get references, so they just left them out to themselves. I had to crop this because like this went quite a long way. So yeah. But it's named some of them, and we found literature that would say that these are also known metabolites of penicillin, which is good because I didn't tell GNPS what organism we got the sample from, but it identified the compounds anyway. Yeah, so the next step, after we got it to work with what should have been quite a relatively easy example for it to do, we moved on to other fungi. And also, my angle on it is co-culture to try and induce secondary metabolism. So antibiotics, antimicrobials often, whether or not a fungi makes it depends on the conditions that you grow it in, so nutrient availability or the presence of other organisms. So we've got other people in our lab who are like growing the same fungi on different media, and they'll find it makes different compounds and it can affect its activity as well. And there's lots of examples in the literature of people using permaculture to try and induce what is successfully induced novel compounds and antimicrobials. So I just adapted the method we had before, so it's still in the fungi in the 24-well plates, but I add in bacteria and broth to the wells along the soil grain outside the fungi. So I'm going to find there's one fungi per plate, and then the first, I've got 4 replicants for each column. So it's the first one's with Acinetobacter, then E. coli, Pseudomonas, Staph aureus, Candida albicans. I'm doing one fungi culture, and then the last one is the fungi by itself. This is a monoculture control. And then one way that we can check if the fungi are active is they can suppress the luminescence of the organisms. So I'm looking just for any plugs or wells where it's dark. So with this particular fungus was not able to stop Acinetobacter growing. But on 1 out of 4 replicants, it stopped E. coli, and 1 out of 4 it stopped Pseudomonas. So it was active against all the Staph aureus samples, which is often what we find. The staph is more sensitive than the other organisms to antimicrobials. And then we filter those, we do SBE, and the next part's basically the same. So when I'm looking at micromograms, one thing I'm looking for is, are there extra peaks in active cultures compared to inactive cultures? So I collect the samples from active and inactive wells separately, and I run them separately through the LC-MS. So in this example, this is just from the diagram detector. There was a co-culture of this fungi with Staph aureus, and I did have more peaks than that sample compared to the inactive one. So that's the kind of thing I'm looking for, and I want to do further investigations on. And also, I'm looking for any peaks that I get enhanced in the co-culture condition like this one, but less in the corresponding monoculture conditions. Yeah, and then you can also do the library searching GNPS, which searches all the publicly available MS2 databases. And when we did the penicillium one, we did find quite a lot of hits, but we're finding as you move on to less well-studied fungi, you don't get as many hits. So often I'm just using 2 or 3, and one of them is the a partner that I just headed to the broth anyway. Yeah, so we do have limitations. We don't have the chemical standards to run for our compounds to get that high level of identification. We don't have an in-house database, so we are relying on these public MS2 databases, and their compound coverage isn't that great compared to just the normal chemical databases. And we also have a bias towards compounds that ionize better in the positive mode at the moment. The thing that we're doing now with it, we're working with George, is to, we've just got access to the NIS20 updates. We're going to try screening our, some of our spectra through there, because I don't think GNPS searches the latest version of this. And I did a quick test earlier, so of the compounds that we found through EMR, of the known ones, about 1/3 of them were in this database. This might give us better chances of dereplicating. But so far, the conclusion is probably we're going to need those large-scale cultures for most of our samples. LC-MS is a complementary technique that can't replace NMR. And ideally, in the future, more people will be uploading their MS2 spectra to public databases like the ones that we can access on the internet. That might be more in the future. Yeah, that's just all the people that have published, which leaves 3 for the LC-MS things that Brent and Melissa are collaborators of chemistry, Susie and Bevan and Lakey, and master students and technicians as well.
A little bit over time, so we have maybe time for only one tiny little question, or we can talk to him during the lunch time in half an hour. Thank you. Our next speaker is Kelly Burrowes. Let me get her.
I was doing that. One person. What if I say? That's what
All right, sorry. Kelly's gonna talk us about the vaping parcel, determining the chemical composition of e-liquids and the resulting aerosol. Please welcome Kelly.
that awkward being the last speaker between lunch, but luckily you've only just had morning tea, so I should be all right. So I'm a researcher at the Auckland Bioengineering Institute, and I'm definitely not an expert in chemistry or mass spectrometry, so that's why we are working closely with Erica and Stuart, and I have my very capable master's student, Angela Yan, who is doing all of the experimental work that I'll talk about today. But what I am an expert in is the overall project that we're working on, so I'm going to start by that didn't stop you recording, that green line. By telling you a bit about the project that we are going to be working on. So this is a 3-year project that's funded by a Marsden grant. So we're only 4 months into this work. And what we are looking at is e-cigarettes. So we're trying to understand the health effects of e-cigarettes. So we found out that cigarettes were pretty bad for people. We didn't personally, but everyone did. So there's been found to be more than 7,000 different chemicals in tobacco or conventional cigarettes. They can be 1 in every 10 deaths around the world can be attributed to tobacco causes. And it's estimated that 1 billion people will die as a result of tobacco-related illnesses this century. And most people realise the effects of smoking on the lungs, so lung cancer and chronic obstructive pulmonary disease, but smoking actually damages nearly every part of your body, so the cardiovascular system, so that's the heart and the blood vessels, as well as the gastrointestinal system, the brain, the skin, and the eyes. So very bad for us. And this is one of the reasons why e-cigarettes, well, it is the main reason why e-cigarettes were invented, to really help support people stop smoking and move away from this dangerous habit. So the current e-cigarette devices are attributed to a pharmacist in China called Han Lik. And these devices were invented in 2003, but actually earlier versions were around as early as 1960. So the big tobacco companies were experimenting with creating these devices, but it was Han Lik who's made the device that we currently know. So it's really, e-cigarettes are really a trade-off, and the biggest problem is that no one yet knows what the long-term health effects are of e-cigarettes, so they haven't been around long enough. So far, cell studies and animal studies are showing that there is going to be some harm from e-cigarettes. So animal studies are showing similar outcomes to smoking in terms of cardiovascular effects, increased chance of lung cancer and emphysema. But I don't think anyone's really going to believe it until we see it in the human population in larger numbers. So really it's just time is going to tell what the answers are. But really there's a trade-off. So at the moment people think, we think that they're safer than cigarettes. So there are less chemicals than cigarettes, of the order of 100 to 200 different chemicals. So hopefully they are safer than cigarettes. So in that sense, they're good for the human population, for people wanting to stop smoking, so transitioning onto e-cigarettes. But on the other side is the younger people or people who weren't already smokers who are starting to use e-cigarettes. And so I've just copied a few media headlines. So e-cigarettes have been in the news a lot recently. And I just wanted to talk on a few of these points. So this one up here, that in the media a lot was this value that vaping is 95% less harmful than cigarettes. So this was actually not based on any real data. It was based on a bunch of people in a room deciding how safe it was. So no one knows how safe they are in contrast to cigarettes. This was e-cigarette and vaping associated lung injury, which happened at the end of 2019, early 2020, being somewhat overshadowed by COVID. But there were nearly 3,000 hospitalizations in the US from e-cigarettes, and this was attributed to a chemical vitamin E acetate, which was added to marijuana containing e-liquids. And one of the main points here is that at the moment, there's no regulation saying that the ingredients in e-liquids have to be labeled. So actually, manufacturers are able to put different chemicals in the e-liquids. All of the chemicals are regarded as safe for oral ingestion, but nobody knows how safe they are to breathe in. And then a couple of articles here. So there's been a lot in the media recently about younger people starting to vape. And this article down the bottom here from the New Zealand Herald a couple of weeks ago was talking about children as young as 10 years old starting to use e-cigarettes. So I personally think it's very worrying how quickly they've been, the uptake has been, and how easy it is to buy these devices. So e-cigarettes work by, they're a battery operated device. So they basically have a heating coil that's either metal or ceramic. So when the device is turned on, the coil heats to very high temperatures. And there's a liquid inside here, so inside this tank, called an e-liquid. And when it's heated, it turns into an aerosol that the user inhales. So this sort of device on the left is called an open tank type e-cigarette. So you can refill it with e-liquids. This image in the middle is called a pod type e-cigarette. And this picture here actually shows that a Juul e-cigarette device. So they're the very popular ones in the US. And Juul created these nicotine salt e-liquids. So they actually mean that you can have very high concentrations of nicotine. So one of these pods, which is really small, about 2 centimeters by 1 centimeter, can contain as much nicotine as a whole packet of cigarettes. The e-liquids, so currently what they do list on the e-liquids, the ingredients are propylene glycol, vegetable glycerol, flavorings, and they can have 0 nicotine, or they can have different concentrations of nicotine. So as I said, at the moment, they don't have to list what these individual flavoring ingredients are or any other additives that they include. So our 3-year project is really, so my expertise is in computational modeling. So really the novel part of what we're doing is bringing our computing tools and methods to bring these different sorts of data related to e-cigarettes together. So the first part that we're looking at is what actually, what's been inhaled with e-cigarettes. So what chemicals are in there, and this is the work that I'll talk a bit more about today. We're also going to be measuring the sizes of the aerosol droplets, and using that data, we want to use our computer models. This is another simulation of our models. We'll use our models to simulate where those particles actually go inside the lungs. So we create these sorts of computational models that are subject specific, so they're sort of anatomically based, so they're realistic shapes of the airways and the blood vessels. We will also be exposing lung cells to the aerosol and measuring any changes that we see in the cells. And again, putting this any outcomes from these experiments into our computer models. And finally, we'll be using MRI or magnetic resonance imaging to image healthy vaping volunteers' lungs before and after they use the e-cigarettes. So we're still looking for volunteers for that, if anyone wants to offer themselves or a friend. So what's currently known about what's in the aerosol? So one of the big issues with this sort of work is that there is a huge range of different products on the market. There's at least 8,000 different flavors that you can get. And each of those different flavors will have different chemicals added to make those unique flavorings, as well as there being different brands which use different additives to make their different products. And there's 3 main aspects that people have looked at and that we are aiming to look at. So the first one is the flavorings. So what flavorings are added and actually found in the e-liquids themselves. So there have been found to be some dangerous flavoring chemicals. One example is diacetyl, but also cinnamaldehyde and vanillin have been found to irritate the lungs. And again, but the long-term health effects aren't known. The second aspect is degradation products. So when you heat that e-liquid, sometimes you get a breakdown of some of those chemicals and forming different products. So ones that have been found in the literature are formaldehyde, acetaldehyde, and acrolein. And the final thing that we're looking at is heavy metals. So some metals have been found in the aerosol, and these are thought to come from the metal heating coil and from any of the sort of parts of the device itself, so soldering material, for example. So our plan is to test at least 20 different e-liquids, and specifically we want to look at e-liquids that are made in New Zealand to really understand the flavourings that are used here. So the new government regulations are going to mean that only specialist vape stores can sell the whole range of flavours. Non-specialist vape stores will only be able to sell tobacco flavour, menthol or mint, and flavourless e-liquids. So we're really focusing on those 3 different products, as well as we've chosen mango as one of the very popular fruity flavours. And we're going to be looking at least 3 different brands and look at how the e-liquids differ. And we're going to be looking at 0 nicotine and 18 milligrams per ml of nicotine. So we've created this vaping robot. So we wanted to be able to create the aerosol and collect the condensate and test what's in there, as well as testing the e-liquid itself. So this vaping robot is automated. So it's really important that we make sure that the e-cigarette is being used in a standardized way. So this is called the vaping topography. So we need to prescribe how long the puff is, the flow rate of the puff, and the pressure that's used. So this device adheres to the ISO standard that's been developed. And this little video shows it in operation. So in here, this is actually, we've got some cells in there, so you can see that we can expose the cells to the aerosol. And in the back here, we've got the condensate collection. So we have a vial that at the moment is in dry ice ethanol slurry, and we try and collect as much of the condensate as we can. So the methods that we are using in Angela's work so far are Headspace, SPME. So we did, in some preliminary work, we tested TDU, GCMS, and liquid injection, GCMS, and we found that we saw the greatest number of chemical compounds using SPME. So we're moving forward using this method. And really what we're looking at here is the flavorings and different additives, as well as looking for any degradation products. We're also using ICP-MS to measure heavy metals. And again, comparing the e-liquids themselves with the condensate samples that we collect. So these are really just preliminary results. So Angela is still working on these experimental measurements, but what we have found so far, this plot here shows the number of different flavoring agents. And we found that actually the e-liquids without nicotine contain more flavoring. Actually, my hypothesis was the other way around, that we'd seen some preliminary work. But yeah, so depending on what flavor you're trying to create, there's going to be a different number of flavoring compounds added. We also found that in this flavorless e-liquid, there actually were 25 different flavorings included. So I'm not quite sure how it can be called flavorless, but I guess these are added to sort of mask the taste of the propylene glycol and the nicotine and the vegetable glycerol. Another thing that's been found in the literature is sort of regulation of the amount of nicotine in the e-liquid. So we actually found in our products that were meant to have no nicotine, they actually did have some nicotine in. And this is another thing that's been found in the literature, very varying amounts of nicotine. So again, there's no regulations in terms of the manufacturing process. There's no standardization of how these e-liquids are created. We also found that the flavouring concentrations decreased in the condensate samples. And I should warn you, there's a very busy plot coming here, so I don't expect you to see all the chemicals in here, but just to really talk through the fact that we're also comparing the e-liquid with the condensate samples. So what we were hoping, I was hoping to see in here was some degradation products, including things like acetaldehyde, potentially formaldehyde. But so far we haven't seen any different chemicals showing up in the condensate samples. So this is the ICP-MS results. So we looked at this table in green here with all the metals that we tested for. And again, these results aren't really what I was expecting, so we will be repeating these measurements. So the hypothesis was that we would not really see any heavy metals in the e-liquid, but we would see some in the condensate. And this is in fact what our preliminary work had shown a couple of years ago. But these experiments that Angela has done has actually just shown, so there is metals in the e-liquid itself. So these could come from the manufacturing process, which is not very well quantified what what metals could come in from the manufacturing or which sort of components they're coming from. But there were a couple of increases in the condensate samples, so cobalt and boron. But our preliminary results shown in this plot down here, we found fairly high concentrations of copper, lead, nickel, and zinc. And these correlate with the metals that are used in the device itself. So the metal coil that we're using is a canthal, so that consists of iron, chromium, and aluminium. So we found some of all of those metals. And then the weldering material, so 95% of the e-cigarettes are manufactured in China, and it's highly likely they still use lead in their soldering materials. So we found in our previous results, we found some lead in the condensate. So like I say, we will repeat those measurements and look into them further. So just to summarize, I really like this poster that's been made by the US Food and Drug Administration. You may not be into chemistry, but if you vape, chemistry may be very into you. So just to summarize what we've found so far, as I said, this work will be continuing for the rest of the year with Angela's masters. So we haven't found any thermal degradation products yet, except for nicotine, so nicotyrene we've found. But we will be really focusing on that towards the end of the year. So it's likely that those products we're looking at are very volatile and we're possibly losing them. So we will be looking at using some different techniques to identify those. Again, heavy metal analysis is not what we expected, so we'll be repeating those measurements. We're going to be looking at some different brands, but really there's just so much unknown about e-cigarettes, so there's a long way for us to go. So that's the end. I just want to thank the Marsden Fund, and we also had a MASH grant to start us off, which was great. Thank you.
Questions? Whoa. Yeah.
How are you measuring particle size distributions?
So we're trying to use the Melvin spray tech device, which is in the chemical engineering department, but it hasn't been very successful yet. So there's been quite a big range of particle sizes. They actually are built to go right down to the nano size. So yeah, I'm not sure if we'll actually get very good measurements, but I always knew that that was the case. So the main thing is that we will use some of the values in the literature for our simulations.
So obviously it falls a little bit beyond, a lot beyond the scope of your research, but have you ever tried, instead of approaching the project in the sense of what's harmful inside the liquid, to see what differences the effect on the metabolites of on the body? So instead of seeing, some researchers have done research on seeing what does smoke actually consist of, but some other researchers have done research to see what changes does it create on the body. And since you are doing metabolites and proteomics, that could be something that will give you more immediate data. For sure, there are going to be some changes before and after, and therefore you could correlate that with smoking.
Yes, so we are in a cell experiment. So a colleague, Bino Suresh, is leading that work. So yeah, I can't remember exactly what we're planning to measure, but definitely we will be exposing cells and then collecting, you know, the material from them. So we could do some measurements in that space.
And for the formaldehyde, I assume you guys try to spike it just to confirm that the method's actually.
We are still in developing that method. We're running at the moment standards of formaldehyde and trying to see if we can quantify it. And but at the moment I don't know. I think that it's in such a very low concentrations in the e-liquids that we loosen it. It's so volatile that so we're trying different techniques. So we need to talk to Udo here. He's the TDU person, so maybe he can give us some devices that we were thinking in maybe when they are collecting the condensate samples, maybe having some of those cells to get those chemicals and inject straight that into the TDU. But we can talk to him later and discuss it. But this is a work in process.
And so, some studies haven't found formaldehyde, and I don't know if that's the methods they've used, or they're losing it, or sometimes maybe it's just not even created. But it would be nice to see some difference in the condensate at least.
When you say cells, is it of human origin or animal?
human, I believe, yeah, but I think, again, my colleague has those, but he's already got some epithelial cells. I can't remember the exact cell line.
So when you use the cells, you will be having those, you don't have a signaling molecules in the intact body. So is it possible for you to include some animal model before you transit to the human clinical trial?
So you mean in a whole, even a whole animal or animal cells?
No, live animals.
Yeah, I thought about that. We're already doing so many different things, but I am collaborating with a group in the US that are doing some animal studies that they've got underway at the moment. So we're sort of trying to learn from what they're finding.
With those degraded compounds you see in the e-liquid, how do you control for what's being degraded in the meat versus what goes through the machine? So I imagine it'll be heated, but it's going through the GCMS.
Yeah, so we're using 50 degrees, right? So I think we're trying to keep it at low temperatures so that we don't get changes in there.
That's it? No more questions? I guess you're staying for lunch? Yes. We can discuss a little bit more maybe with some food. I would like to thank all of our speakers. It was a very great and interesting morning, such a different topics and all using mass spectrometry. So it's just to show the main purpose of this symposium is to show the community what it's available, but also the applications of these techniques into many different research studies. So thank you all for coming, and I hope you enjoy lunch, and we see you later for the afternoon session.
Just a very quick note on the lunch. If you are not already vegan or gluten-free, please don't become today, because we have very special dietary requirements, yeah, for people that have chosen to.
Thank you. Thanks.
So